Thank you for those presentations. That's wonderful. So at this time, I would like to welcome Dr. Sherman, Dr. Bondi, and Dr. Jordan and my co-moderator, Dr. Wang, to the panel discussion. As a reminder, we encourage our audience to submit questions and comments using the chat box located below the webcast video. Please include your full name and your affiliation with your questions.
So those were wonderful talks. Dr. Sherman, I will start off with you. In terms of your discussion about data going into registries, since today focuses on pediatric cancer, you know, the National Cancer Institute has a major childhood cancer data initiative, which is really trying to get structured data from patient records, including genomic data or even just typical clinical genetic markers into a structured format. Is there any attempt to sort of combine that data with registry data? You know, you talked in your talk about trying to have more molecular data, and that could be a source. I can't hear you.
Dr. Sherman, are you we cannot hear you. Can you double check your microphone?
Why don't we give you a minute for that question? And Dr. Wang, did you have a question for one of the other, because my other question was also for her, so.
Yeah, sure. While Dr. Sherman sorted out her microphone situation, we do have a couple questions from the online audience. So I think one thing that one of the questions from the online participant, Ruth, she asked, are the demographic and or social determinant of health, should we consider them as effect modifier or rather than confounders? And how would that affect ability to detect the environmental exposure and the cancer in the cluster? So.
Thanks. That's an excellent question. And I think social determinants and other factors could be both confounders and effect modifiers, because it depends upon different types of exposures they might have and predisposition and other types of social, where people live and those kinds of factors. So I think we have to look at it in multiple ways to evaluate whether it is an effect modifier or a confounder.
Yeah, thank you. That is a very, very important aspect of the thing that we certainly would like to incorporate in the analysis. Do we have Dr. Sherman here that can follow up?
I think she may be left to try to reconnect.
Okay. Yeah.
Dr. Jordan, let me ask you a question. You talked really eloquently about genomic or genetic ancestry, but the computer programs or the algorithms to assess that keep changing dramatically with regard to your ancestry 500 years ago versus your ancestry in early in human development. And is there some way to sort of integrate those different layers of genetic ancestry into your analysis?
Yeah, that's a very interesting question. And your question kind of touches on two areas. One is this current state of the art with respect to the algorithms for inferring ancestry. And then the other one is this something that we don't, we think a lot about that, but we think much less about this temporal aspect that you mentioned, right? Because when you talk about ancestry, you can characterize ancestry at different time strata. So I think the first question is a little bit easier to answer. And the answer to that is that the algorithms are actually quite good, and the state of the art with respect to algorithm development is pretty stable. But what can change with respect to your ancestry, and anyone that's done 23andMe or Ancestry DNA knows this, is that those proportions that you infer can change based on the reference populations against which you compare your genome, right? So when we infer ancestry, we're comparing our genome sequence or the genome sequence of the study participants that we're looking at against global reference populations. The algorithms that do that work really, really well, but they're only as good as those global reference populations. So fortunately, that representation has gotten better and better and better over the years. And so we are constantly refining and improving our ability to infer ancestry at different levels of relatedness, at the continental level and at the local level. So that's looking good. Now, with respect to this temporal aspect, that's a very interesting and a little bit more difficult problem to unpack, right? So when we infer ancestry, at this time, we're sort of thinking about two global epochs or timeframes. One is the modern timeframe where we have global travel, lots of migration, very cosmopolitan, very diverse. And then there's the sort of pre-global travel era going back, 500 or 1,000 years. And so when we, again, infer ancestry now, either in our scientific studies or with those programs like 23andMe, we're looking essentially at that at that distinction, right? We're asking where our ancestors came from 500 or 1,000 or 2,000 years ago before the era of global travel. But you have posed an interesting challenge whereby you can go back even further and look at distinctions between more ancient hominids, right? So one of the very interesting things about the ancient DNA revolution is now that we're seeing admixture from non-modern human species like Neanderthal and Denisovan. And so, but again, that depends less on the algorithms and more on the availability of the reference data against which we have to compare. As that improves, we'll be able to do a better job of inferring ancestry across different time strata.
Great. Okay. Dr. Sherman is back. So let's see if we can get your audio working. Yeah.
Can you hear me now?
Yes.
Yes,
Okay.
wonderful.
My apologies. So I think your question was about the National Childhood Cancer Initiative that's going on with NCI. And so NASER is involved in a portion of that. So we are working on a National Childhood Cancer Registry where we are combining the state level data into a national data set. Generally, the national data sets that are representative, covering the entire population, don't, outside of the state, when you move to the national level, you don't generally have the PHI, you don't have a name, you don't have other information that would be important to link to additional cohorts. So this particular registry is a little bit different, and there are authorities in place to be able to collect the more specific information so that that registry data can be linked to other to exposure data or genomic data or whatever data is available and approved through a mechanism. So yes, so cancer registry data is an important component of that larger project.
Go ahead, Dr.
Yeah, just to follow up. So Dr. Sherman, can you maybe comment on, whether the CDC and or NASA envision a future where the state registry data and or the variables can be better supported and detect cancer clusters, and how exactly would that happen? What information are still needed that is currently not available?
Well, I can't speak for CDC, but I will say that I personally am an internal optimist, and that is what a lot of NASER's involvement in these projects are really to move and to leverage the cancer registry data more effectively. So I think a lot of the speakers so far this morning have talked about lack of data or lack of merging of the data. Some of them, obviously the cancer registry doesn't collect any of the exposure data or any of the genomics data. And so linkages, like we've been talking about, could be the next step, and we are moving in that direction. Also, we've talked a bit about latency as well. And so trying to tease out exposures and latency over time requires residential history information. And so we don't have that place information over the history of a patient's life yet in the cancer registry data, but we are moving in the direction we have the structure to collect that information. And the SEER, the NCI cancer registries are already involved in collecting that historical information. But I also think that one of the best ways to improve how cancer registry data can help inform cancer cluster investigation, maybe not so much inform cancer cluster investigation, but to really go to that next step where we're improving the health of communities, is to not have these investigations as a standalone. The information that comes out of the studies is very important to move the scientific field forward, but we should not wait for cancer control or other mitigation activities to come sequentially, but rather to occur in tandem and connectedly.
Thank you.
Dr. Bondi, given that you're at Stanford, a center of AI and machine learning, there was a question about, given the difficulty of perceiving these clusters, are there attempts to use machine learning approaches to try to more rapidly identify them in existing data sets?
Yeah, I saw that question by Dr. Perdue, and I think it's really fascinating to work with the registries somehow to be able to look at clusters. And there's a lot of epidemiologic studies that are being done looking, just looking at the data by residents, and also combining it maybe with electronic health records so that we would have better informed data sets with where people live, and trying to look at these clusters. And one of my former postdocs used the registry data looking at exposures and race, ethnicity, and I think it's a way of then identifying where clusters could be and then going back in and trying to work in those communities, or to look at exposures that could have occurred in those populations. I think we really have that opportunity now to be able to do that.
I'd like to add something quickly to that. I was also intrigued by that question, and I think there's certainly tremendous potential for machine learning and artificial intelligence approaches to define those clusters. And for Dr. Perdue, one of the interesting things you might want to look at is this concept of explainable AI, because up until the last few years, the direction that artificial intelligence and machine learning was going was towards these black box methods, where it was difficult or impossible to understand how individual features were contributing to the clusters or whatever classes you define. But now there's this explosion of techniques in so-called explainable AI or explainable machine learning, whereby you can define clusters using high dimensional data with lots of interactions, but then you can open up the black box and unpack those clusters and figure out how the specific features contribute to the clusters, which is a very important part of trying to tease apart what's causing the clusters that we see. So explainable AI is a really exciting development there.
Yeah, thank you. I want to circle back a little bit in terms of incorporating genomic or biomarker information to the cancer registry. So Dr. Sherman mentioned about their efforts, sort of the virtual pool cancer registry linkage system. And I'm wondering, and also, one of the questions from the online audience asking about single cell sequencing or other more recent development in the sort of genes expression analysis. Is that platform, virtual linkage platform, can be adapted to incorporate this type of technologies, with the data generated from this type of technologies? You are muted.
Operator error, not technical problem there. One of the advantages of the VPR is that it is certainly more quick to respond to advancing science than the collection of data through the cancer registry system. So the VPR is set up so that we have a linkage process in place where we link the cohort patient information with the cancer information, and then anything that is associated with that cohort in terms of data would be then merged together. So even experimental or uncertain information about the relevance certainly can be included in that way. So as long as we have an identifier, we generally focus on name, date of birth, social security number. Those are the ones that we're able to link to. But the other way that potentially could work, of course, is a lot of what we're talking about today is place. So we can link disparate information by the location. So the cancer registries do geocode cases down to the block group level. So if there's specific information that's locational, there's the potential to link it that way as well, although this genomic information is unlikely to be in that context, but that's another avenue.
Yeah, thank you. We have Stephanie Foster would like to make a comment from the CDC.
Thanks, I appreciate it. I just wanted to follow up on the question about adding additional information into the registry systems. And as Dr. Sherman has eloquently explained, this is something that I think we all are hopeful for moving towards in the future. And I think it links very nicely with one of the comments from Dr. Alexandrov in the first session that there's a lot of research that's still needed while the genomic markers have been that we're working towards incorporating genomic markers and understanding the genomic markers of cancer and environmental exposures in established clusters. That work has not really been done yet. And so it's exciting to look forward to the work that can be done in that area. I wanted to also mention that, thinking sort of long term as well, if you look at the Woburn example that was brought up, while initial investigations were not able to identify a known cause, it took many years, I think almost 2 decades, before that relationship was established and confirmed between the TCE and PC exposures and those childhood leukemias. And so I think that that's an important lesson learned, that these investigations might necessitate long-term commitments to evaluating what those relationships are, and as the science evolves, what we might be able to understand. Thank you, I appreciate it.
Thank you, Stephanie.
So there was a broad question, which maybe I'll ask Dr. Bondi, since you are not a member of a health agency, but from the epidemiologic perspective, so let's say an individual calls of the public and is concerned about a cancer cluster. What is sort of viewed as sort of the minimum actions that a public health agency should do?
So, I've had calls over the years, and, being an epidemiologist, not having access to as much data as a public health agency that has access to all of the registry data, I always refer them to the registries. Because I think for an evaluation, they always do some kind of an evaluation. It may not be the best, but has been the responsibility of the registries or the public health entities to work with the community and the individuals who report that concern.
So then just to follow up on that, Dr. Sherman, my own experience working with registries, which have been a great help to our research, has been more retrospective. It's not in the moment data. So because you're accumulating data all the time. And so how do you handle if there's a report of a cancer cluster now, let's say 3 children were diagnosed with cancer in the neighborhood in the last year? How would the registry go about sort of identifying additional information and things like that in the moment?
So because we're population-based, our data is, I hesitate to use the word delayed because we can't get the population data any faster, but to really support cancer cluster investigations, we need to have the data on everyone in the area, right? So you're exactly right. We would be looking at retrospective data, but there's also mortality data that tends to come out a little bit faster than the cancer data, but there's just less variables attached to that. there's a minimum data set initiative that's we're working on through NASER, where we're trying to identify what's the minimum set of variables that we can collect more quickly to be able to respond to. For instance, one of the uses might be cancer cluster investigation. So we might be able to get some data a little more quickly. But by the time a cancer has been reported or a potential cancer cluster has been reported to the registry or to someone else, it has already occurred and is generally already in the data set. So we obviously do have models for future rates and trends, but we aren't able at this point to apply them to any sort of local area in terms of trying to predict cancer clusters proactively that way. But the more proactive analysis, rather than waiting for a community concern to come up, which often represents a community that has resources, is a much more equitable way to try to identify and research cancer clusters, where we're actively looking at the data and not relying on someone who's able to identify which person to call at the cancer registry. So in that sense, that portion of the new guidelines, when we're in a position to be able to implement that, will really serve the community better.
And perhaps just final question on that to Dr. Jordan on that, since the topic here is molecular or genetic characterization, is there sort of a list of recommended samples to collect for molecular analysis if a cancer cluster is? Or in your mind, is there a set of samples you could imagine that would be most useful to subsequent research?
Sure. And just to clarify, by samples, I think about the kind of data we generate. Are you talking about the tissue samples or the kind of data we want to generate, or perhaps both of those?
Sort
right?
of both.
All right. So, I'll just throw out a wish list here, right? So take this with a grain of salt. So with respect to samples, the gold standard, what we would love to have, if at all possible, is matched normal and tumor samples, right? Now, that's not always going to be possible, but that will allow us to do comparisons between the normal state of the tissue and the cancerous state of the tissue, which would be just great if we could do it. In terms of the type of data, I'm a geneticist, I'm biased, so of course I want DNA data. And again, the matched data is really key and important there. So we want to be able to characterize the germline variants, those variants that we are born with, as well as the somatic mutations that arise as a part of the tumor genesis process. So genomic data, I think that epigenetic data, at the very least DNA methylation data, is fantastic. And what we've seen in our own work is that a lot of the group-specific differences that relate to cancer health disparities are actually epigenetic rather than genetic differences. So that's quite important. And then finally, information on how genes are expressed. Again, with the key being, can we measure differences between expression levels of genes or proteins in normal versus tumor tissue? That would be, again, kind of a wish list, but the baseline of the minimum kind of data that we would need to be able to do a really good job of looking at the relationship between multi-omics and cancer.
Yeah, thank you. We probably can take one more question from the online audience, maybe just in general, a question asked about segregation, redlining, and some additional new type of risk factors maybe need to be considered in the cluster detection analysis. Is there any analytical approach that currently has been proposed to incorporate that kind of information in the cluster detection investigation?
Yeah, I think that's quite relevant to health disparities. So if it's okay, I'll take that one, at least at first. I saw that question too, a very interesting one. I think first and foremost, the answer to that question is yes, that's certainly a part of what's going on here. We know that social determinants of health are very important to cancer. And one of the things that I'm quite excited about that I hear from Dr. Sherman is this connection between geocoded data and then the molecular, again, multi-omics data that I just talked about. So with geocoded data, where you live, place-based determinants of health, sometimes called your ZNA for the zip code. There's a tremendous amount of information that we can glean from geocoded data about structural inequities and structural determinants of health. So I think the key there with respect to looking at the legacy of discrimination as it relates to health disparities is to tie that geocoded data together with the genetic and other multi-omic data.
Yeah, thank you. Thank you very much. I think we are right on time with our panel discussion. So I would like to thank all the panelists for a great discussion and great presentations. I think now we are going to have a 15-minute break, and we will return at 11:30 a.m. with session 3 on the topic of environmental exposure and the pediatric cancers. Thank you very much, and look forward to see everybody back in 15 minutes.